Thank you all for coming. Today's seminar is by Dr. Frederick Ekuban. And Dr. Ekuban is an assistant professor in the Department of Medicine in the Division of Gastroenterology, Hepatology, and Nutrition. He received his Bachelor of Science with honors from Kwame Nkrumah University of Science and Technology in Ghana, and then a master's in general toxicology and environmental health risk assessment from the University of Eastern Finland, and then a Ph.D. in pharmacoscience and toxicology from Tokyo University Science Noda campus in Japan. So really, Dr. Ekuban is quite an international scholar. And during his PhD work, he published one first author paper in toxicology, which is a premier journal, and eight co-author papers. So he clearly was quite productive in the lab. From November 2021 to March 2023, Frederick was a postdoctoral scholar in the Department of Pharmaceutical Sciences at the University of Tennessee Health Science Center in Memphis, and there he published 2 co-author papers. And then he joined UofL as a postdoctoral fellow in Dr. Matt Cave's group in April 2023, and he thrived here under Dr. Cave's mentorship, as have many other people. And I'm so glad that Frederick was able to receive that mentorship and training. He published one first co-author paper about alcohol and PFAS and how that impacts liver disease in mice, and also 2 co-author papers with Matt's group. He's received a number of honors while in Matt's lab, including being named a Kentucky Colonel. He received the PFAS Awareness Gubernatorial Award from the United Nations Association of Kentucky. He placed first in the postdoctoral poster presentation at the 2024 Ohio Valley chapter of the Society of Toxicology meeting, and at the National Society of Toxicology meeting, he placed 3rd place in mechanism specialty section. In June 2025, Dr. Ekuban became an assistant professor in medicine, and he was named a presidential research scholar in the Louisville Clinical and Translational Research Center in July. So he clearly has ascended in the ranks. He's a member of CIEHS, and he serves on the CIEHS Career Development Program as representative to the Internal Advisory Committee of CIEHS. Dr. Ekuban's long-term research goal is to establish a robust program investigating how environmental pollutants modify other disease-associated risk factors with an emphasis on molecular mechanisms, obviously a topic near and dear to my heart and to those hearts in the room, and identifying biomarkers of susceptibility. And he wants to develop evidence-based interventions to reduce environmental health burdens and improve public health outcomes. So today he's going to talk about his research and I think his plan as he moves forward. So thank you very much for speaking to the group today.
All right. Thank you so much, Dr. Klein, for the very generous introduction. All right, so I'm very excited for this opportunity, and I thank the organizers and faculty leadership for inviting me and giving me this opportunity to give this talk. This afternoon, I would want to share with you a story about how hidden modifiers, as far as alcohol-associated liver diseases are concerned, exist, and to what extent do they influence the effects of alcohol-associated liver disease spectrum. And so I would first walk you through a few backgrounds regards to environmental pollution and delve deep also into non-communicable disease, spectrum of diseases, and we would also touch base on alcohol-associated liver disease, and then bring you back to the environmental pollution and introduce you to the PFAS story. And I will then walk you through some few highlights about the work I've been doing, looking at some human studies, and then some animal model studies, and few key takeaways, and would open the floor for your comments and discussions. And the learning objectives, as have already been shared, I'm going to walk through describing the variability in ALD disease progression. We're going to explain how environmental pollutants could interact with alcohol, leading to the worsening of liver injury, and then look at some mechanistic insights, as well as some translational implications of these environmental lifestyle interactions. So I would always want to start with a story that I always love to use in explaining the concept of toxicology. Imagine being in a room like where we are now, filled with all kinds of people, having fun, from the elderly all the way down to small kids playing, and just having having very good time. And all of a sudden, this bad guy just enters the room, the tiger, and look at how ferocious it appears. What are you going to do if you were in such an environment? Can see here, just as we would do, people just running for their lives. Some are trying to take shelter. Some are, trying to run as fast as they could. Some want to hide, and all sort of chaotic events. But more importantly, as I did indicate, this room is filled with different classes or different levels of people. And so we do have older adults, we do have younger adults, we do have nursing mothers, pregnant women, and people with existing health conditions. And so their ability to run away from this threat, as in the tiger, will not be the same. For example, I think these young kids here might not even know what exactly is going on. Like, why is mom and dad, running and shouting, and so in dealing with the tiger in the room, it requires several approaches. First, we need to identify the threat, and several questions have to be asked. First, to talk about the characteristic features, the behavior, how does the tiger attack? And this will be referred to as the hazard identification. Secondly, we need to understand the situation fully well. How did the tiger enter the room? Was the door fully locked and the tiger broke it loose and entered the room, or the door was actually left open and the tiger found its way easily in? These questions will help us also establish the exposure assessment by understanding the situation fully. Thirdly, we need to determine the level of risk, as in, is this really a serious threat to life? So let's use this as an example. For example, relative to the tiger, coming into the room, how about let's say a cat entering the room, or how about a dog entering a room? Will there be that much of panic as we saw with the tiger? And so understanding the level of risk will help us to characterize the risk. And then also we need to develop control strategies. That is, how do we keep the tiger at bay and make sure that everybody is safe, the kids are safe, unharmed, the elderly are safe, unharmed, and what kind of controls would actually work best in this scenario? And that will constitute management of the risk. Then lastly, just like we are in here, majority of people are outside, this building, or outside in their labs and in other places. And if we were the ones encountering this threat, we should be able to disseminate that information for people who are not in the room and might not have not even known what exactly was going on, to be aware that, look, there is a tiger in town. And so everybody should stay indoors, or, be very careful when you are going out and all that. And, and so this form of communication will help, communicate the risk to various people at large. Again, we need to understand, just as I talked about earlier, there are vulnerable populations, and we do have them in different levels of sensitivity. So, for example, the children have bodies still developing. They are not even aware of exactly what is happening. Pregnant women can, they are carrying their baby in the womb, and they don't have the full strength to run away and escape from the threat easily. The elderly, who might even have underlying illnesses or conditions, health conditions, might not also be very strong enough to run away from this threat of a tiger. And so all these forms of vulnerabilities should be well understood and well characterized. And lastly, like workers, in the context of the general, assuming that someone's duty is to work in this place over, a certain long period of time in the day and encounter this threat. So how about I then tell you that this story about the tiger that I shared is just a metaphor to represent the actual situation of having environmental pollutants within our neighborhood, within our homes, within our environment. And just like the same scenario where you had different types of people, adults, young and old, pregnant women, and others, having these environmental pollutants in the midst of all these people also constitutes a threat. On a daily basis, we do have a lot of chemical tigers, so to speak, in our environment that we face. Most of these, some are natural, some are anthropogenic, and for example, from industrial sources to automobile exposures of fumes, and then domestic activities, we are encountered on a daily basis with several of these chemical tigers. These environmental pollutants have been basically categorized as either chemical pollutant, physical pollutant, or biological pollutant. And there are several subcategories of these. And today, for the purposes of this meeting being an endocrinology centered, I don't think I'll be allowed to go out of this door if I don't touch base on endocrine related pollutant. And so we'll talk a little bit more about that. And on a much broader scope, these compounds or pollutants have been divided into found in pharmaceutical products, personal care products, among others. So therefore, humans are on a daily basis exposed to a complex mixture of chemical substances ranging from several lifestyle activities to occupational activities, transportation, and then the natural sources, as I did talk about earlier on. And also thinking about the water we drink. And at the end of the day, these pollutants entering the food chain, getting biomagnified in the food chain, and at the end of the day, humans getting exposed, in very high quantities. And so, what are endocrine disrupting chemicals? I would share three definitions by three different regulatory authorities. First, from the US NIGHS, which define endocrine disruptors as natural or man-made chemicals that may mimic or interfere with the body's hormones, which are known as the endocrine system. And these chemicals are linked with many health problems in both wildlife and people. The World Health Organization, working together with the International Program on Chemical Safety and the United Nations Environmental Program, have also defined endocrine disrupting chemicals as exogenous substances or mixtures that alter functions of the endocrine system and consequently causes adverse health effects in an intact organism or its progeny or populations. Lastly, the European Food and Safety Agency, EFSA, also defines an endocrine disruptor as an adverse effect in an intact organism, an endocrine activity, and a plausible causal relationship between the two. So endocrine disruptors have a very long history, way back to the 20th, early to mid 20th century, where a lot of activities related to the evolution of endocrine disrupting chemicals started. And so it all started somewhere in the mid 20th century, like around 1920, where in the US, some farmers reported infertility in pigs, which were fed some mold grain on the fields. This also was followed by another report of infertility, this time around in sheep grazing on specific clover in Australia. And so it continued. And between the 1950s and about getting to latter part of the 1990s, there were several reports of endocrine disruption in wildlife across the globe resulting from environmental chemicals. Here in the US, in 1971, NIEHS studies show that diethylstilbestrol which was essentially prescribed as a drug to control loss of birth or or let me say miscarriage, was later identified as a transplacental carcinogen. And the progeny of those who suffer from this cancer also suffer other non-cancer related conditions. And so it went on and on with the publication of the famous Silent Spring book in 1962, and then having the Wing Spread Conference in 1991 here in the United States. And the author of the book, The Silent Spring, after publishing her book in 1962, passed on somewhere in 1964. And so a group of other authors came together and also as a follow-up from this Silent Spring publication, also made a second publication related to being aware of exposures to these chemical pollutants, Our Stolen Future, published in 1996. And it went on 2009, the Endocrine Society issued a statement. And actually, I did forget to say that the Wing Spread Conference was the first time that the term endocrine disruptor was proposed and started to be used. And then in 2013, World Health Organization with the United Nations Environmental Program prepared a very comprehensive report about environmental pollutants as far as the endocrine system concerned. So a few of endocrine disrupting chemical sources, and they are found in everyday consumer products, personal care items, and also occupational exposures. And so, for example, bisphenols in feeding bottles and toys for children, and also phthalates in cosmetics, perfumes, and others, and PFAS which we'll talk about just in a bit, found in non-stick cookwares, firefighting foams, fabrics, carpet, among others, and a whole lot of other types of endocrine disrupting chemicals that have been identified. There are about greater than 1,000 known endocrine disrupting chemicals, and about well over 50 of them are known to be obesogens. So a few mechanisms of action relating to these endocrine disrupting chemicals and their pathophysiological pathways. Most notably, these endocrine disrupting chemicals are able to mimic the hormone receptors or different hormones, and they're able to compete with these hormones in their specific sites of their specific sites of action or specific site of attachment. And in that case, because these specific hormones have been denied access to their regular functions, it has its own downstream dysfunctional pathology or pathogenesis related to that. And also, there are some of these endocrine disrupting chemicals that are able to block these hormones and even prevent them from being able to function in the first place. And so here, in terms of pathophysiology, you can look at the various targeted organs, looking at the reproductive and endocrine, the brain, the pulmonary and cardiovascular, and the different types of EDCs that are responsible for these. Health effects are numerous, ranging from in children, conditions of related to the lungs like asthma, hyperactivity, HD, in women, cancers of the breast, in men, thyroid disorders, and also prostate cancer, among others. And there's been also a very good correlation that has been established as far as the developmental origin of health and disease is concerned. The fact that exposures encountered during the formative stages of life in the fetus, all the way through the lifespan of the person until, later life of adulthood. It's been established to see a very good trend of how these endocrine disrupting chemicals are able to affect lifestyle in adulthood, which were actually exposed during their formative stages. So non-communicable diseases remain a significant global challenge. And to date, it's been predicted that about 70% of death worldwide are often caused by these non-communicable diseases. And most of the time, it's been noted that these are actually avoidable causes. And environmental exposures alone constitute about 23% of this 70% incidence. Root causes for this mostly have been physical inactivity, unhealthy diet, environmental exposures, and then harmful use of alcohol. And so today, these are the two that I'm going to delve a little bit more to show you how research is evolving to show the interaction of these root causes of non-communicable diseases leading to their fruits, so to speak, of very bad disease outcomes, heart diseases, chronic respiratory diseases, diabetes, and among others. Lifestyle factors include things like physical activity, nutrition, tobacco use, alcohol consumption, and sleep or rest. And to start with, alcohol consumption represents a very significant public health challenge globally. And it's been established that about well over 130 million disability adjusted life years, DALYs, are caused by the misuse of alcohol. There's also about 3 million deaths every year as a result of harmful and excessive use of alcohol globally. And here in the United States, excessive use of alcohol is shown to cause about 95,000 deaths annually. All these are as a result of a spectrum of diseases, which collectively is referred to as the alcohol-associated liver diseases. This disease spectrum normally starts with a buildup of fat in the liver, which is referred to as steatosis, and then progresses to a form of intense steatosis with some inflammation, referred to as steatohepatitis, which could then also become fibrous. And at this point, it's if there is still continued consumption of heavy amounts of alcohol, then the liver will now become cirrhotic. And at this point, it's irreversible, which would require transplant. And sometimes and occasionally also about 3 to 10% can also result in hepatocellular carcinoma. However, recent epidemiologic and clinical data has shown that not every heavy consumer of alcohol results in severe alcohol steatohepatitis or alcohol liver disease. And it's been shown that about 35% among a given population of heavy alcohol consumers suffer from severe ALD. The 65% do not really develop any such serious effects from that. This establishes the fact that there could be several other contributing factors beyond the consumption of alcohol, such as epigenetics, genetics, gut microbiome, sex as a variable, and then environmental exposures, which we are going to also talk about. So this really has established the concept of what we call the two-hit hypothesis or multiple hit model. And this hypothesis essentially establishes the fact that most liver disease progression is influenced by multiple insults. And so there should be a first hit, which would cause some baseline outcomes. But for the disease to move on to a much more severe pathological levels, there should be a second hit, which could be any of the factors I did mention earlier, and that results in severe liver disease and sometimes goes into cirrhosis or even hepatocellular carcinoma. So among a group of chemicals that we talked about, which humans are on a daily basis exposed to, these group of chemicals referred to as per- and polyfluoroalkyl substances, PFAS for short. And interestingly, these are also types or examples of endocrine disrupting chemicals, as I showed earlier on. These PFASs have been grouped into different categories. I'll touch on whether as a non-polymer or polymers. And with the non-polymers, they are further divided into either perfluoroalkyls or polyfluoroalkyls. And let's focus on the perfluoroalkyls, where we do have two different structural PFASs. The first is PFASs, which have, and so I must say that these PFASs expresses, or they do have carbon-fluorine bonds, which are known to be one of the very strong, most strong bonds in chemistry, and that is one of the reasons why these substances are known to be very resistant to breakdown in the environment. And so beyond their carbon-fluorine bond, there is a terminal functional group. And these functional groups could be either a carboxylic functional group or a sulfonic acid functional group. And so here you can see this one, perfluorooctanoic acid, which is PFOA, having a carboxylic acid terminal functional group. And then perfluorooctane sulfonate, PFOS, having a sulfonic acid functional group. And with time, they realized that there were several health effects with these legacy PFASs, and so they were phased out and mostly banned, and there were alternatives to these. However, these alternatives also have been well researched into, and research has shown that they also have serious health implications. And so there are these new generational PFASs like Gen X that have also been produced. And to date, although these are relatively new, research show that they also have serious health effects. So today we'll touch a little bit on one example of the PFASs with a sulfonic acid functional group, which is the PFOS. Uses of PFASs include several others, several like I did mention on earlier on, microwave popcorn bags, non-stick cookwares, fast food containers, and then by way of exposure at the occupational level, the use of firefighting foams and retardants. So PFAS on its own, just like any other PFASs, have been also well characterized. It's been shown that they are very significant environmental and public health pollutant as a result of their widespread presence and bioaccumulation. In humans, they accumulate for about 4 to 5 years, and in rodents about 37 days. And last 2 years, International Agency for Research on Cancer classified PFOS as a Group 2B possibly carcinogenic compound. Health effects have, for PFASs have ranged from different organ systems, from cardiac through reproductive to kidney and liver damage. And I also wanted to make mention of specific PFAS effects on the endocrine system. And endocrine organs like thyroid, reproductive tissues, pancreatic organs, and then in child development as well as in pregnancy, PFASs have been shown to cause several levels of toxic effects on these organ systems. So why do we need to worry, or why should we be concerned about this scenario? PFAS from this map here shows a widespread, presence across the country, and especially towards the east, where there are a lot of industrial activities showing even much more higher presence over there. And in recent times, there was a publication which revealed that these PFASs could be found in beers within the US. What they did was they took about 23 different beer products across the country and analyzed it for PFAS content, and 22 out of these 23 were found to have PFAS levels that were beyond the exposure limits that have been set by EPA. And most notably were BS found in North Carolina, where PFAS has been shown to be a big issue, especially around the Cape Fear River Basin area. I would just run through this effects of PFAS, and lifestyle activities like diet have been reported, showing neurodevelopmental effects in animal models. And then also recently a group of researchers have also shown that PFAS together with high-fat diet drives prostate cell metabolism into prostate cancer. Now, the big problem here is that whereas independent effects of PFAS exposure and alcohol consumption are well known, there isn't much clarity about their combined effects or the interactions, thereby leaving a huge data gaps, limited understanding, and lack of comprehensive kinetics or dynamics as far as PFAS and PFOS in this context is concerned. And so we, in my lab, and also as a carryover working with Dr. Cave, we first developed a study which involved human alcoholic patients, and then followed it up with animal model study. So we took blood samples, plasma samples from patients who had already been classified into various severity of alcohol steatohepatitis, and we sent their samples for what we call the exposome analysis. And exposome analysis is able to tell the extent of chemical exposures that an individual has encountered over the lifetime. And following this, we also did an animal model study where we used an animal model of alcohol-associated liver disease and exposed concurrently to PFOS for 10 days. And then on the last day, gave a bolus of ethanol, and then euthanized the mice after about 5 to 6 hours. So what we realized in the human study, in brief, was that we saw correlation of various PFAS levels to various severity of alcohol steatohepatitis. PFOA and PFHPA showed significance in this correlation. The others did not, but you could clearly see a very clustered correlation between the levels of PFAS in the plasma of these patients and relating alcohol steatohepatitis severity. We did a lot of pathway enrichment analysis, and we found several terms. It's a bit, the texts are a bit smaller, but we did find very interesting terms related to several pathways that explains life cycle pathways, metabolism pathways, among others. So the previous one was for PFOA. This we also did for PFOS, and also did find interesting pathway changes. And then we did the MWAS for alcohol steatohepatitis itself, and we're able to also find very similar pathways in terms of vitamin, protein pathways, bile acid pathways, and other metabolic processes. In the animal models, what we realized was that PFOS exposure exacerbated the alcohol-induced steatosis and liver injury. And so here you could see that whereas alcohol alone generated some amount of lipids in the liver. With the introduction of PFOS, this was even much more increased. And we're able to corroborate that with oil red O stain for that. And then also liver to body weight ratio was increased. And then as well as plasma ALT levels, which is a marker of liver injury, was also significantly increased in this combined PFOS with alcohol group. We then did some lipid analysis, both in the plasma and in the liver, and we did see that triglycerides were consistently increased in plasma, and same with the liver. Also, we did find a similar trend with VLDL levels also increasing. Cholesterol, however, in the plasma was decreased by alcohol consumption. However, in the liver, alcohol increased the level of cholesterol, but with the introduction of PFOS, this was brought down back to almost basal levels. And also we saw that alcohol in general had an effect on non-esterified fatty acids. We also did measure blood alcohol concentration levels, and we found very interesting data here, which we consider more to be very interesting data relative to what we had seen with the liver toxicity, as mentioned previously. We saw that PFOS significantly reduced the blood alcohol concentration levels and significantly also upregulated the various ethanol metabolizing genes and enzymes like CYP2E1, ADH, and then the ALDH. And we're able to reproduce this by running some protein expression analysis, which also showed very significantly increased protein expression of CYP2E1 and ALDH1A1. And then we did some analysis relating to mass balance calculation. And by mass balance calculation, what we are trying to do here is that we first want to ascertain how much of the pollutant was given to the mice and how much was distributed, and then how much was eventually excreted. And so when we did that analysis, we realized that the liver, regardless of alcohol consumption, had the most accumulation of PFOS. And it's interesting that most of the metabolism also occurs in this same liver where the PFASs were shown to be at their highest. And we did some RNA sequencing analysis, and basically what we showed here was that when we look at comparisons that establish ethanol alone effect, which is the ethanol vehicle versus ethanol fed PFOS, and juxtapose that with the ethanol alone effect, the PER-fed vehicle versus ethanol vehicle, and juxtapose that with the effect of the combined group in terms of the number of differentially expressed genes that were established, we did see that there were commonalities and differences. And so with only ethanol, we see genes like heme oxygenase 1, FGF21 being upregulated. But when we expose PFOS together with ethanol, those genes that were upregulated with ethanol alone were downregulated. Again, we also saw that CYP4A12, which is a target gene of PPAR-alpha, for which PFOS and majority PFASs are known as key agonists for, was also downregulated with ethanol. But with the introduction of PFOS and alcohol, they were upregulated. And so these were also very interesting result that we got from the transcriptomic analysis. We also did several pathway analysis and did find pathways that consistently were related to fatty acid lipid metabolic processes. And then processes that were predicting the long-term exposure consequences of alcohol and PFAS, including liver cancer, hepatic steatosis, and fibrosis of liver, among others. Lastly, we also did expression, qPCR expression of key nuclear receptors and their target genes. And interestingly, we found all of them to be significantly upregulated by PFAS and alcohol. And then last but not least here, we did find that PPAR alpha was predicted as the top transcription factor that was predicted to mediate the effects of alcohol PFAS interaction that has been shown. Very interestingly, we tried to look at the human data and the mouse data, and I'm so excited that we were able to find several overlaps. For example, linoleic acid was found as a common pathway in the humans, MWAS with PFAS, and MWAS with alcohol steatohepatitis, and in the mice data as well. And so this is very refreshing, knowing that what we are doing could be translated towards human relevance. In summary, we were able to establish from this study that PFAS exposure enhances alcohol metabolism. It increases buildup of fat. It decreases the body weight, and then increased plasma and hepatic lipids, as well as increased ALT levels. And there were other mechanisms that underlie relating to upregulation of lipogenic genes, nuclear receptors, and other enriched transcriptional pathways. In conclusion, we show here that this study provides very novel insights, and then also establishes the fact that PFAS exposure may increase the risk of promoting ALD. And this altogether establishes the fact that there is a need for targeted interventions which could help mitigate these adverse effects. And so ongoing studies in my lab now is to look at the exact role of PPAR alpha as a transcription factor or as a nuclear receptor, and its influence on the PFAS alcohol interactions, and also look at translational findings by using a humanized PPAR alpha mouse model. And also we're looking at the structure activity relationship by using other types of PFASs like the carboxylic acid functional group and the alternatives to see how different types of PFASs respond with alcohol. It's also very interesting that we've also started preliminary studies to see how dietary fiber could help in trapping the PFASs within the human system and processing it for easy excretion. And we are collaborating with one of our researchers collaborators in Boston University, who has very interesting preliminary data on this. And then also expansion of the human exposome analysis. So this study was published recently in Toxicological Sciences, and the UofL News made an article for it. You can take time to read it whenever you have the chance. And also, Dr. Cave and I were giving an in-studio interview at KET to talk more about this. And then what we have done is an addition to a few that has already been done, looking at dichloroethane and ambient particulate matter, Gulf War illness chemicals, and PCBs. These are a few models of environmental pollutant that have been used in concert with alcohol consumption. And I was thrilled to see that Gallup polls shows a decreasing trend for a very long time in the consumption of alcohol. And I don't know whether people are getting more sensitized and educated about studies like what we are doing. All right, so I just want to remind you back to my tiger that there are a lot of PFAS tigers that remain and that we have to face on our in our daily lives, and therefore we have to stay alert and remain vigilant and work together to tame this ferocious tiger. To this point, I want to say thanks to my mentors, Dr. McLean, Dr. Cave, and collaborators, and want to pause here for your questions, comments, and discussions. Thank you very much for your attention.
They covered so many things. That was just a fabulous talk.
Thank you.
So much. So, were there any changes in retinoic acid or retinoid levels in the liver in your study?
We've not looked at that yet, but it's something that we want to expand more to look at other pathway areas. So it will be good to take a look at that. Yeah, so yeah, that's we didn't
It
do.
was a short-term study.
Yes, it was a short-term study. So and this was the first time we were trying to do PFAS model with alcohol, and so hopefully towards future studies we will try and incorporate investigations like glucose tolerance test and all that. But in this premier study, we did not look at that, but we did measure the level of glucose, and we saw that alcohol had a significant decreasing trend on glucose levels. But it will be interesting to, plan that for future studies in the course of time.
Seems like it would be kind of complicated, difficult to give the PFAS to the mice in a way that might be similar to environmental exposure.
Right.
So, and I didn't follow exactly how you said it.
Yeah.
But gave him some, and then I think in a couple of days.
Yeah.
So, I'm wondering, and then I wonder also, so, and then do those levels and the mice reach some kind of a level that you might expect from environmental exposure. These seem to be very complicated.
Right.
Calculations to make.
Yeah. Thanks a lot for the comment. So there are different types of exposure modules. In this very case, we did use PFAS exposure by oral gavage. And so because in this model, we really wanted to, in as much as trying to model the human scenario of alcohol consumption, we wanted also to study some of the mechanisms that are involved with this PFAS combination with alcohol. And so the issue of the dose level, we used 1 milligram per kilogram in this case, which is a little bit above the usual human recorded exposure levels of PFAS, which are almost on the nanogram scale. But however, given the fact that we're doing a very short-term, exposure studies of 10 days, we wanted to give some reasonable level of dose that could elicit some mechanistic pathways that we can really describe. However, towards the future, we plan to do a much more longer exposure studies for about, let's say, about 12 weeks of exposure. And in that case, we are planning to use environmentally relevant doses of PFAS or different types of PFASs in conjunction with the alcohol.
And is there any information yet on the exact mechanisms of it does that? To tell you the truth, I'm not even sure exactly how alcohol causes the initial liver disease.
Right.
Red, you know, it has oxygen, reactive oxidized species.
Yes.
Like that, but I don't have good control.
Yeah. Preliminary.
By all the research I've read about it.
Yes, the preliminary data has shown that one of the key mechanisms is by the activation of nuclear receptors. And nuclear receptor pathways, we saw tremendous upregulation of several different types of nuclear receptors that are known to play a role in lipid metabolism, xenobiotic metabolism. And so and that's why we want to continue this study using the PPAR alpha knockout mice, that we can study the role of PPAR alpha and other supporting nuclear receptors. And it will be very mechanistic to see how these turn out from this kind of exposure in those mice. Yeah, thank you, sir. Yes. Yes. Not really. Yeah. Yeah. So it's very surprising for us actually to have seen that much of levels of PFAS in the liver, and we also did measure PFAS in the urine and the feces. They were very low, like as you could see from that data. And so it's not very clear at this moment the exact mechanisms related to how long they could stay in the liver, or when how we could even be gotten rid of from the liver. So I believe that going forward with the upcoming studies and investigations, we'll be able to close in on that. And one, I was just talking to Dr. Klein, I'm preparing an R21, which we are centering it on looking at the metabolism of alcohol by PFAS. And we are proposing to do some temporal measurements post the bolus bench. So we're going to euthanize the mice at certain point times, like 0.5 hours, 1 hour, 2.5 hours, and then at 6 hours. And these can really help us relate the causality of things that we saw to the PFAS exposure, and at what exact time point we are seeing these changes. And so hopefully in about, if all things go well and that my grant is funded, in about a few months to years, I'll be able to get you very good answers for your question. Yes. Yes. Contrary to what is already in literature, that these PFASs are not metabolized, in this study, we saw a lot of metabolism activity and so many different types of cytochrome P450 enzymes being upregulated, even also in the, yes. Yeah, so that is what we don't know at this moment. But judging from the way there were so many metabolism activity, it's plausible to infer that possibly these PFASs are getting metabolized some way, somehow. But notably, just recently, there was a study published showing that the gut microbiome has a potential effect on being able to metabolize certain types of PFASs. And so I've already reached out to some of the investigators for collaboration on how we can expand this study to look at the microbiome and PFAS metabolism and how it results in the overall effect by way of explaining some of the mechanisms involved.
Is it more likely that it's metabolism that's leading issues?
Yes, that's a very interesting question. Especially with the alcohol result, what we saw was that PFAS, for example, decreased the blood alcohol concentration levels, which in essence also increased the metabolic rate of alcohol. However, paradoxically, we still saw, severe toxicity in those mice. And contrary to the opinion that lowering of blood alcohol levels should reflect in low level of toxicity, but paradoxically, even though PFAS was regulating this metabolic activity, we still saw, so that's also part of my, that's also part of my R20 grant to expand on this and find some answers why PFAS on one hand is increasing metabolism of alcohol, but at the same time and paradoxically resulting in increased toxicity. Yes, sir. Yeah, that would be interesting. So there is an investigator in our department who does MET ALD. That's a new term for obese conditions with alcohol, and we are also about establishing a new collaboration on that front to see how obesity in the midst of these pollutant lifestyle interactions could also yield any potential effects. Yeah, and so it's interesting. I'm very excited that a lot of people have reached out to me to collaborate. There is a new investigator who got into the PharmTalks program department, whose investigation deals with pancreas and the general endocrine system. And he's very interested to find what happens to the pancreas and the entire endocrine system. So I guess we have a lot of very interesting answers to provide along the way. And hopefully we'll be able to gladly share that with you, in the course of time.
Just if there are any questions. So there was a question about their gender differences.
Yes, thank you for the comment. So in terms of the gender, actually, when we published this paper, that was a huge critique that we received from the reviewers, like, why are you doing this study in only males? And we explained that because this pollutant and alcohol interaction model is much more novel, we wanted to use a model system that has been much more reported as far as alcohol-associated liver disease was concerned, and help to establish a background fundamental data that could be expanded on. And so that's one of our next steps, to include females in this same exposure module and see how females' response differ from or might be the same with the males. And so that also, I'm unable to give an answer for that for now, but hopefully in subsequent rounds, when the opportunity is given, I would be able to share more updates on that. Yes. All right.